seminars I go to, nobody claps at the beginning, so that's really kind of... All right, thank you all for coming. I appreciate it. So I've been here now about 4.5 years, and you know, as was mentioned, my laboratory's research interest is in breast cancer biology, and really we have 2 or 3 main areas of interest, one of which is breast cancer genomics. But the other has always been, for many years now, the biology of a particular subset of breast cancer, the so-called triple-negative breast cancers. We're going to say a little bit about that term in a minute. And I chose to talk about that and focus on that today because of the emphasis of this seminar series on racial disparities in cancer development. And this is certainly the type of breast cancer, and as I'll show you, that really drives the disparity in breast cancer mortality. And it's really a very important public health problem and clinical problem in breast oncology. And I think our lab is making some inroads in terms of how to begin to treat that and approach that differently. So that's going to be the focus of my talk today. So let's talk a little bit about these subsets of breast cancer first. So traditionally, breast cancers have been classified very simply by the presence or absence of 3 proteins that tend to be expressed or not on breast cancer cells. The estrogen receptor, the progesterone receptor, and those 2 often go hand in hand, but not always. And then a 3rd protein called the HER2 or ERB2 oncogene. And so traditionally, we've talked about hormone receptor positive breast cancers that are ER and or PR positive, or HER2 positive breast cancers. And then that leaves this sort of subset of breast cancers that don't express any of those 3 things. So they're ER negative, PR negative, HER2 negative, and this is where this terminology of triple negative breast cancer comes in. But it's really a terrible term, right? Can you imagine trying to describe someone to someone else and saying, well, the person you're looking for is not tall, is not blonde, and doesn't wear glasses? Not tremendously helpful. And that's the case with these classifications as well. And as a result, we're really moving away from this terminology and really tending to talk more about breast cancers as either being, now the pointer's dying, either being of the luminal subtype or the basal subtype. So those subtypes are shown here. So what you can see is about 73% of all breast cancers sort of fall into this luminal A category, and there are about 12% here in the so-called triple negative. This isn't really very helpful. So what we're doing now instead is really classifying them based on this 50 gene set, so-called the PAM50 gene set, which allows us to look at intrinsic subtypes in a clinical setting, really, and tells us a lot more about the biology of breast cancers. And so here is this bar graph here shows the breast cancers that normally would have been referred to as triple negative. So they're hormone receptor negative and HER2 negative. And now these are categorized by this PAM50 designation of expression profiles and cell types. So what you can see is that about 75% or so of the triple negatives fall into this category of basal-like breast cancers. And so a lot of times people use these terms interchangeably. It's almost correct, but it's not quite correct. So whereas most triple negatives are basal, you can see that there are some triple negatives that fall into this normal-like category, some that are actually HER2 positive, and others that are either luminal A or luminal B. And then by contrast, if one looks at these hormone receptor positive, HER2 negative, the so-called ER positive breast cancers, about half of them are luminal A, about half of them are luminal B, but you can still find some basals even in this setting. So this is the classification based on this gene expression profile as either basal or luminal is much better. It's a much richer way to describe the biology of breast cancer. And it has an impact and it has an influence on prognosis and disease outcome. And so we've known now for a long time that patients who develop luminal A in particular kind of breast cancer do best. So this is overall probability of survival over a long period of time. And the luminal A breast cancers are, first of all, they're the most indolent. They progress very slowly, so even patients who do ultimately die of their breast cancer take a long time to do that. When we identify patients with luminal A breast cancer, we follow those patients for 15 years because they can recur even up to 15 years after the initial diagnosis because the progression of the disease is so slow. By contrast, if you look at the basal-likes and maybe the HER2 enriched, these are the two most aggressive types of breast cancer, you can see that this curve begins falling off very rapidly. So we actually have a good idea for these patients that within 5 years of diagnosis how they're doing, whether we've achieved a cure in these patients or not, because the story is pretty well told within 5 years, in contrast to the luminal A's, where we really have to wait 15 years to really make some confident predictions about what happens next. So clearly then, the basal breast cancers are more aggressive, and they have a higher mortality rate than the luminals. And again, as we move forward in this classification scheme, we're even moving away from luminal A versus luminal B. And there are those in the field who just who really just promote the idea of just classifying breast cancers as either basal or luminal and leaving it at that. There's a lot of information there. So the basal breast cancers are very aggressive. They progress rapidly, and they're quite therapy resistant. This is important because many people feel that it's the basal type breast cancers that drive the disparity with regard to mortality in breast cancer. And so if you look at the proportion of these different types of breast cancer in non-Hispanic white women, African American women, Hispanic women, Asian Pacific Islanders, you can see at a glance the difference between the luminal A's and the basals. And this, if you look at these results here and you account for these differences, it's become pretty clear that the preponderance of basal-like breast cancers in African American women and the reduced prevalence of luminal A breast cancers in African American women really go a long way to accounting for this difference in mortality. So again, as many of you probably know better than I, the incidence of breast cancer among black women versus white women, black women incidence is a little bit less, but the mortality rate is significantly higher. This is about a 10% increase in breast cancer mortality. And again, many of us believe this is accounted for by the lack of prevalence of luminals and the increased prevalence of basals, again, these being so much more aggressive. And again, this is a probability of survival curve. So this really leads to two very important scientific questions, only one of which I'll talk about today. And the first one is, why are African American women more likely to develop basal breast cancer than other ethnic or racial groups? There's obviously, when we talk about these sorts of issues, there's two general possibilities. It's genetics, genomics, or it's environment. We certainly have a lot of people who are looking at genetic influences of disease susceptibility. We have some of those folks doing nice work on that here. And I think it's very likely that there will be genetic components to risk of different types of breast cancer. But of course, we know that lifestyle factors, behavioral factors influence cancer risk as well. And so I'm sure this is going to be a sort of a complicated mix of genetic and environmental factors that drive this. I don't work on that, so I'm not going to talk about that anymore today, but I'm going to talk more about what our lab focuses on, and that's this question. Why are these breast cancers more aggressive and more deadly than luminal breast cancers? And why don't we do better at treating them in the clinic? Because it's one thing to have a cancer that's a type that's aggressive versus not, but there are aggressive cancer types that we actually treat very effectively clinically. And that's not the case for these breast cancers. They're not only aggressive, but they tend to be drug resistant. And that really has a huge impact on how these patients do clinically. So we're going to focus on today on some of the work that my lab is involved in that's really aimed at addressing this question. So I'm mindful of the fact that this audience is a mixed group of population scientists and molecular and cell biologists. So I'm going to sort of try and walk a line down the middle there, and we'll see how we do. Okay, so to study this question, we use a panel of breast cancer cell lines that my laboratory developed back in the 90s called the SUM breast cancer cell lines. And there's 10 of them shown, and all of these are derived from different patients. There are some cell lines derived from primary tumors, from different metastatic sites. And they represent the sort of the spectrum of basal and luminal breast cancer cell types. And the ones I've shown here in red are these so-called basal breast cancers. And today we're going to focus really on two of these cell lines that we work on a lot, one called SUM149 and one called SUM229. SUM149 was in fact derived from an African-American woman with aggressive inflammatory breast cancer. Again, both of these patients did not do well, had very aggressive disease, and ultimately died of their breast cancer. So we're going to spend a lot of time talking about these two cell lines today. And one of the things I just want you to focus on, because this is going to come up in the talk, is that this gene here, PTEN, is an important player in the story, not because of its presence, but because of its absence. So PTEN is one of those genes that becomes really important in cancer development when it goes away as a result of some genetic alteration. So if these cells are PTEN negative, and again, as you'll see, we think this is a very important aspect of their biology and why they're aggressive and why they're drug resistant. Okay, so then I'm going to use as a control for a lot of the experiments I'm going to show you a cell line called MCF10. A lot of people don't know this, but MCF stands for Michigan Cancer Foundation. That's where I did my postdoctoral fellowship, and I actually work with the guy who developed this cell line as well as the MCF7 breast cancer cell line. These cells were derived from a reduction mammoplasty from a patient with fibrocystic disease. They're spontaneously immortalized, so we can grow them forever in culture, which is really helpful for being able to do these experiments. They are of the basal epithelial cell type, so they're a really nice control for basal breast cancers. We're going to talk a lot about this gene, the EGF receptor, epidermal growth factor receptor, because as you'll see, this plays a very important role in basal breast cancer. And these MCF10 cells are EGFR positive, and they actually require signaling from this protein in order to proliferate and survive. So in that way, they're very similar to the basal breast cancers, but there are important differences, as you'll see. And so this is an outstanding control cell line for all of the experiments we do with these basal breast cancer cells, the SUM149, the SUM229, and others. All right, so I want to talk a little bit about the epidermal growth factor receptor for a minute, because it represents one of the most frustrating aspects of basal breast cancer. One of the reasons that the triple negative tag stuck around for so long is because if you have hormone receptor positive breast cancer, we can treat you with a hormone receptor targeted drug. If you have HER2 positive breast cancer, we can treat you with a HER2 targeted drug. But if you don't have any of these, we don't have a targeted drug for these patients. And that's frustrating because these cells, as I said, express to high levels the epidermal growth factor receptor, EGFR, and we have a drug that attacks that. It's called gefitinib, or there's another drug called erlotinib. It's very effective clinically in other settings, but not in breast cancer. And so that's really frustrating as to the presence of this receptor, which is an important signaling molecule for these cells. The fact that we have a drug that really is good at knocking out its activity, but it doesn't work in the clinic. So the EGFR is a transmembrane protein. Here's the cell surface. Here's the outside of the cell. Here's the inside of the cell. These proteins are activated by growth factors or ligands that bind to the ectodomain here and cause the receptors to form these dimers. And once they're formed as dimers, they're now active enzymatically, and they phosphorylate a bunch of sites down here, which is critical for the transduction of their signal. The important thing for this talk, though, is what's going on up here, because almost everything we know about the epidermal growth factor receptor comes from studying its cognate ligand, epidermal growth factor. Stanley Cohen actually won a Nobel Prize a number of years ago for discovering EGF. And so if you do a PubMed search of EGFR, you're going to find like 50,000 papers, and 49,000 of them will have to do with the ability of EGF to activate the EGFR. But what I'm showing you here is that EGF is only one of the players. And in fact, there's a whole family of growth factors that can activate this receptor. And in breast cancer, EGF is of no importance at all. Rather, what's important in breast cancer, particularly basal breast cancer, is a different growth factor called amphiregulin. And that's what we're going to talk a lot about today. Amphiregulin binds to the EGF receptor, causes it to be active, and sends downstream signals. But its biology is very different, and it causes the biology of the EGF receptor to be very different. And we believe that has a lot to do with this peculiar biology of these basal breast cancers. So we're going to talk a lot about amphiregulin as an activator of the epidermal growth factor receptor. All right, so here's a little cartoon that shows a little bit about amphiregulin, its domain structure. I'm sure you don't care about that. But what I can show you here is that when this protein is synthesized by the cell, again, here's the cell membrane, here's the inside of the cell, here's the outside of the cell. The full-length protein gets sort of stuck into the membrane like this. There's a protease called Atom17 or TACE, which comes along and clips right here, and that allows these soluble molecules to be secreted. And they come in different molecular sizes, and I'll show you an example of that in a minute. But the important point is once these are clipped and secreted, they can turn around and they can bind to the EGF receptor and turn it on and drive the signal. Now, one of the reasons that we think this is really an important cancer growth factor is because of where this protein is normally expressed in normal cells. And it turns out that AREG is normally expressed during puberty in the mammary gland, and is also expressed during development in embryogenesis and the development of the kidney and the lung and other organs that make branching ductal structures. And it turns out that amphiregulin is really essential for the organization of these three-dimensional tissues, for penetration of these cells into stroma, for matrix reorganization. EGF doesn't do any of this, but amphiregulin is really good at it, and this is why we think it's a good cancer growth factor, because it really drives some of the important phenotypes that cancer cells express that they're not supposed to. So we're really have been excited for a long time about the biology of amphiregulin. And one of the first things that we discovered when we were looking at this is that amphiregulin, again, fundamentally changes how the EGF receptor behaves. So these are our MCF10A control cells. Here are some 149 cells. Again, some 149 cells are always making amphiregulin. They're making it all the time, dumping it into the medium, and we've proven that secretion of amphiregulin is required for these cells to be able to grow. So we did a very simple experiment. We did an immunofluorescence experiment looking for where the EGF receptor was localized in these cells. And one of the things that caught our eye at first is that when you have these MCF10A cells that have EGF in the medium, because they need it, and you look for the EGF receptor, it's all in these vesicles, and a lot of these vesicles are actually around the nucleus. See it better here. By contrast, when you look at the SUM149 cells, the EGF receptor is all at the cell surface. There's a good picture here. You can see them here. So this is a hugely different cellular localization of EGFR that's determined by the ligand that's activating it. And in fact, if we take MCF10A cells and we just take all the growth factors away, the EGF receptor will come to the cell surface. But then if we give it either EGF or amphiregulin, within 30 minutes, all the EGF receptor is inside the cell with EGF, but with amphiregulin, it stays out here at the cell surface. And we can actually turn MCF10A cells into SUM149s by making them express amphiregulin, and then this is what happens to the receptor. And what we found is that when you do these experiments, again, the biology of the receptor changes dramatically. So it's been known forever, almost forever, that in normal cells stimulated with EGF, that the receptor is internalized and then degraded. So what you can see in this experiment is a stimulation with EGF, and you can see that the amount of receptor that's present in the cells is rapidly going down. This is part of the normal biology of the EGF receptor. The ligand activates it, it signals, the cell brings it inside, cuts it up, turns it off, right? So this stuff is always turning over. But that is not what happens when you give the same cells amphiregulin. Rather, it builds up, it accumulates, and in fact, it accumulates at the cell surface. And so, so we really feel strongly that this is a significantly different biology because of the receptor signaling from a different compartment, and it's living a lot longer. And in fact, we know from clinical studies that when you see this sort of pattern of EGF receptor on the cell surface in clinical breast cancer specimens, that's an indicator of bad prognosis, aggressive disease, and poor outcome. And we've known that for an awfully long time. And so our results indicate that this mechanism, the presence of amphiregulin, is what really drives this EGFR overexpression. So amphiregulin is a game changer in terms of how it influences the biology of its cognate receptor. Okay, so that was pretty exciting. And so, you know, in the 21st century, when we find a protein that we're interested in and we want to study it more, we knock it down. We shut it off using shRNA or, more recently, CRISPR-Cas technology. So a number of years ago, we took our SUM149 cells and we used shRNA technology and we knocked out AREG and we developed a bunch of different clones that had different levels of knockdown of the protein. And you can see that here. So I remember, before I mentioned that the protein comes in some different molecular weight forms. So you can see them here. And this is this clone number 3, or clone number 4, and a pool of shRNA. So the bottom line is these cells no longer express amphiregulin. So we were able to really get rid of it quite effectively. And sure enough, when we did, these cells really slowed down in their proliferation. So that was encouraging. But what was even more interesting to us is that when we get rid of the amphiregulin, the cells lose their ability to penetrate matrix and invade. And of course, invasion is a key aspect of the aggressiveness of cancer. It's an essential aspect of metastatic spread. And so this is an invasion assay where you put the cells on one side of a matrix, they penetrate through, and then you stain the other side. And you can see that the normal MCF10A cells don't invade. The breast cancer cells, SUM149, and a control cell line that got a shRNA but a non-silencing one, invade very nicely. But when we shut off the amphiregulin, these cells not only don't grow, but they lose their ability to invade. And this, again, is very consistent with the biology of this growth factor in terms of where it's normally expressed in developing tissue. So we think this amphiregulin EGFR axis is not only important as a driver of proliferation of these breast cancer cells, but as a driver of their aggressiveness and their invasiveness. So that was pretty exciting. So now we know that AREG is different from EGF, and in terms of how the cell behaves, in terms of where it is in the cell, in terms of some of the phenotypes it influences. But we really don't know very much else about mechanistically what's going on. So we did this really cool experiment using something called a reverse phase protein array. And we took our MCF10A cells and our SUM149 cells. And the beauty about working with these 10A cells is we grow them under defined serum-free conditions, and we can give them whatever growth factor we want. So we can give them EGF, or we can give them amphiregulin, and they behave differently depending on what we provide for them. So we did this experiment where we culture them in the presence of either EGF or AREG. We have our SUM149 cells, which are making their own AREG and stimulating themselves through this autocrine loop. And then we have this cool drug, gefitinib, that allows us to turn off the receptor under both cases, and then follow that over time and see what kind of signaling molecules are influenced. And so when you do this really cool experiment, you get results that look like this. Don't panic. Within this mess of beautiful colors and tree view diagrams, there's some really cool data that we can extract out. I'm just going to show you a couple pieces of that. So one of the things that we looked for in this experiment, we looked for proteins that were upregulated either in MCF10A cells grown in the presence of amphiregulin and the SUM149 cells, but different from the MCF10A cells grown with EGF. So we're looking for things now that are really about amphiregulin and not really about whether the cells are breast cancer cells or normal cells. And you can see that we found a few interesting things here, but I want to focus on this one that just jumps out at you. So this is a protein called fibronectin-1. And this result suggested that just taking these MCF10A cells and taking out the EGF and giving them amphiregulin caused this huge increase in the expression of fibronectin-1, and that was validated here in these experiments. So this is just a Western blot showing the levels of fibronectin-1 in SUM149 and SUM229 in the 10A cells grown with EGF, and here's what happens when you give them amphiregulin. It goes up, and this is protein level data. This is messenger RNA level data. So again, 10As with EGF, 10As with AREG. So AREG actually turns that gene on and causes it to be expressed to high levels at the messenger RNA level, which is then translated into protein, which we see here. So this is pretty exciting because fibronectin and its receptor integrins really are what mediates this invasive capacity. So we really feel strongly now that the ability of amphiregulin to drive the upregulation of fibronectin-1 is a major, major aspect, a major factor in the ability of this signaling axis to drive this phenotype of cellular invasion. So this was a really exciting connection between AREG, fibronectin-1, and the ability to invade. So when we get rid of AREG, the invasive capacity goes down. So we're pretty excited about this finding. And in fact, there are drugs in development that really specifically block the interaction between fibronectin-1 and alpha-5 beta-1 integrin. And actually, a colleague and friend of mine at Michigan, when I was there, developed a drug called ATN-161. It's been through phase 1 clinical trials. It's got tremendous preclinical activity. It's sort of stuck now because of the company that decided to license it and disappeared. But we have it in the lab, and we're very excited about moving forward with this drug because, again, it specifically blocks the ability of fibronectin-1 to activate integrins and we think drive invasion. So it's a potential drug target that actually comes from these experiments. Now, the other thing I just want to point out, because it's going to come up again, and I already mentioned it, is that in the SUM149 cells, if you look here, this is SUM149. This is that gene I told you about, PTEN. It's essentially not present on this array experiment, and in fact, confirmed by Western blot. PTEN is completely gone in these two breast cancer cell lines. We now know very clearly that the loss of PTEN is a common feature of highly aggressive basal breast cancer cells. There was a really nice clinical study done at Vanderbilt a number of years ago, actually just a couple years ago. And it was a study in which patients with triple-negative breast cancer were enrolled in a neoadjuvant chemotherapy study. So in neoadjuvant chemotherapy, patients get diagnosed with their breast cancer. They had their chemo before the surgery. And you can then follow the tumor over about 6 weeks before the surgery. And it turns out that in some patients, the neoadjuvant chemotherapy gets rid of the primary breast cancer. It's all gone. Now, those patients still get surgery. But then in other patients, the neoadjuvant chemo shrinks the tumor. And in other patients, there's no effect. And you can imagine that that effect of neoadjuvant chemotherapy is predictive of what's going to happen later. And patients who have a poor response to that chemo end up doing badly and recurring. It turns out that patients with the kind of basal breast cancer where PTEN has been lost had a 0% complete response rate to neoadjuvant chemotherapy. 0%. Not one patient in that group had a complete pathological response. So again, another very important clinical connection between the loss of this gene and drug resistance. And again, drug resistance is the name of the game. I don't care how aggressive these cells are. If they're drug sensitive, we'll kill them all and the patient will be fine. So this is really an important aspect linking this part of the pathway to how they respond to drugs. Okay, so we have another don't panic slide. All right, so one of the central dogmas now in aggressive breast cancer and other cancer types is that one of the reasons that the therapies are not very effective is because within the population of breast cancer cells, there is a subpopulation of so-called cancer stem cells, and that the cancer stem cells themselves are A, responsible for the recurrence of tumors, but B, are intrinsically drug resistant. And one of the things that we've known for many years in breast cancer is that this pathway, the so-called Wnt signaling pathway, is an important player in breast cancer biology. We are absolutely sure about this. The problem is we have no idea how. We still don't understand how it works. But all of the data suggest that Wnt signaling plays a role in stem cell biology. It certainly does in development. And probably it does in cancer biology as well. So we became very interested in this Wnt pathway, and I'll show you the connection to amphiregulin here in a second. But I just wanted to show you a couple things about this pathway. There's a lot of really funky names to the proteins involved in this, like frizzled and disheveled. And this all comes from the fact that these proteins were all discovered by fruit fly biologists who, if you work with fruit flies, I guess you've got to think of clever names for your genes because you're working with fruit flies. Anyway, so I'm showing you two pictures of this pathway. In this picture, the pathway is in the off position, and in this picture, the pathway is on and signaling. And there's just two particular proteins I want you to focus on. One is called DKK1, and the other is the Wnt itself. So the DKK1 is the factor that turns the pathway off, and the Wnt is the signal that turns the pathway on. When the pathway's off, this receptor here is not signaling, and this protein complex that's called the beta-catenin destruction complex gets formed, and when it gets formed, this beta-catenin molecule goes to the proteasome and it gets cut up in pieces, and so beta-catenin levels in the cell stay very, very low. But when the Wnt pathway's on, this destruction complex is disabled. Beta-catenin levels now build up, and they actually go into the nucleus, interact with a transcription factor, and drive expression of a whole bunch of target genes. And so beta-catenin, so there are a number of key players in here: beta-catenin, disheveled, DKK1, Wnt, and so forth. When we looked at our protein array data that I showed you a minute ago, we got pretty excited, because what we found is that in the SUM149 cells, the levels of this protein called disheveled were increased. The overall levels of beta-catenin were increased. The levels of GSK3 beta were decreased, but its phosphorylated derivative was increased. So this, to us, looked like a signature telling us that Wnt pathway is active in these cells. It's like, well, that's pretty interesting. But we needed a way to now see that biologically. This is nice circumstantial data suggesting the pathway must be on, but how do you actually visualize that in the cell? So what we did is an experiment where we took a reporter construct that would light up with green fluorescent protein in individual cells in which the Wnt signaling pathway was turned on. And we looked for those green cells in cells that we normally grow in two-dimensional culture in monolayer and culture dishes. You can see that very, very little was going on, which was a little disheartening. But when we took the same cells and we grow them in so-called stem cell media, in conditions in which they make these three-dimensional tumor spheroids with factors that promote the development of stem cells, suddenly we get these clusters of cells lighting up like Christmas trees, indicating that there are groups of cells in these clusters that have very active Wnt signaling. So that was pretty exciting, because it suggested it was consistent with this evidence, and it suggested there was some pretty active Wnt signaling in these SUM149 cells. So that was pretty cool. And we had this other data that had been lurking in the lab for a while, and now suddenly everything started to make sense. I mentioned about this DKK1. This is the molecule that turns Wnt off. And one of the things we found is that amphiregulin significantly suppresses DKK1 expression in MCF10A cells. So here's that data here. Here's the MCF10A cells without any growth factors and have a lot of DKK1. If you give them EGF, and it comes down a little bit. But if you give them AREG, it goes down even more. So that was pretty interesting. So then I had mentioned that we did these experiments where we knocked out amphiregulin. And so we looked at the relationship between amphiregulin and DKK1 in these cells. And so these two bar graphs here show the level of AREG being made by the cells, and this shows the level of DKK1 being made by the cells. So here are the SUM149 cells, lots and lots of AREG, very little DKK1. And again, we had noticed this inverse correlation in some of our microarray data. And when we started to now knock down amphiregulin, DKK1 levels started going up. So here's, now we knock down AREG, here's DKK1. In this particular clone, AREG is low, DKK1's really high. And this one is sort of in the middle, where we didn't get very good knockdown of AREG, and not much happened at the level of DKK1. So we have these 3 or 4 now sublines where we know something about their ability to make amphiregulin and their ability to make DKK1. And we took those cells and we put them back in mice to ask if these cells could now continue to form breast tumors in these animals. And what you can see is that in the SUM149s and in a control treated with different vectors, that these cells make tumors quite aggressively in vivo. The C1 clone now, this is the one here where we didn't reduce amphiregulin very much, and DKK1 cells remain low. These cells were a little bit slower in their ability to make tumors, but they still made them. But this is really exciting, the C2 clone, again, where now AREG is low, DKK1 is high, and these cells don't make breast cancer in the mouse at all anymore. We've never been able to see a tumor in animals. We've done this experiment several times. You always get nervous about totally negative data. It's like, oh man, somebody killed the cells, or somebody spilled them on the floor, or all kinds of crazy things go through your head. But we're pretty convinced that these cells have completely lost their ability to make tumors. And again, this sort of brings together this connection between AREG and EGFR and Wnt signaling and the ability to make tumors in vivo. So we're pretty excited about this and continue to hopefully move forward with this work because AREG itself becomes a potential therapeutic target. One can generate antibodies that there are plenty of antibody drugs out there that target growth factors, and so it's conceivable to actually target the amphiregulin itself. So the first summary here is that these highly aggressive triple-negative or basal breast cancers drive autocrine activation of EGFR via this growth factor amphiregulin, and this differs substantially from EGFR signaling. We believe regulates motility invasion via this fibronectin-1 integrin signaling pathway, and regulates stemness and tumorigenicity via this Wnt signaling pathway. And this is all about amphiregulin, so that's a lot going on by this one factor having such a dramatic influence on the biology of its receptor. Okay, another don't panic slide. So I'm going to shift gears a little bit and talk about what else is going on in these cells that we think is cooperating with the amphiregulin. So I've now talked a couple times about this protein PTEN. So this is the PI3 kinase pathway, the phosphatidylinositol 3' kinase pathway, blah, blah, blah. The important thing to remember for this talk is that this enzyme basically does one thing. It takes this molecule in this cell membrane called PIP2, and it adds a phosphate to it and turns it into PIP3. And PTEN takes it in the other direction. It's a phosphatase. It takes PIP3, it clips off the phosphate, and turns it back into PIP2. So we know that this is a very important signaling pathway in cells. Once the cells make PIP3, they activate a couple of other enzymes, this one called PDK1, this one called mTOR. And the target of these two is another protein here called AKT. And we know that AKT it's got a long, sordid history in cancer biology. It was present, originally discovered in an oncogenic retrovirus. The gene gets mutated and amplified and activated in all kinds of cancers. We know this is an important player in cancer. But what's interesting is that we have a drug that blocks this. We have drugs that block this. We have drugs that block that. And none of them work in this setting. And everybody would think, and everybody thought, that when you have these breast cancers where now PTEN is gone, like I showed you, then this pathway is turned on. There's lots of PIP3. There's lots of this. There's lots of that. And so surely to goodness, these cells ought to be sensitive to the drugs that target this pathway. Not so fast. And so one of the things we're interested in is why these cells don't respond to these drugs. All right, so now I'm going to introduce a different cell line that goes, in addition to our favorite SUM149 and SUM229, a cell line called MDA468, also a basal breast cancer cell line, also PTEN negative, also EGFR positive. But the difference is that in the MDA468 cells, the EGFR is active by a totally different mechanism. The gene is amplified, copy number amplified. It's active without any ligand at all. So the difference between MDA468 and these is amphiregulin. No amphiregulin here, lots of amphiregulin here. All right, so now we did a simple experiment. Well, Erica did this experiment. It's not that simple. It looks simple, but it's hard to do. Take the 468 cells and put back PTEN and see what happens. And what happened is exactly what you'd expect. When you put PTEN back, this levels of AKT phosphorylation just dramatically go down, right? This is exactly what you'd expect if this is regulating that, and we can read it out by looking at AKT. If you put PTEN back, you'd expect the phosphorylation of this to go down, and in these cells, it's just beautiful. It just goes away. Unfortunately, when Erica put PTEN back in SUM149 and SUM229, nothing happened to AKT phosphorylation. I mean, that's really, the difference between this result and this result is pretty striking. So that was puzzling. So then we started working our way through the pathway. So here's a drug called BKM120. BKM120 is an inhibitor of the enzyme that does this. the PIP2 to PIP3 conversion. It's a very good drug. It works very effectively. And when you give BKM120 to either the MCF10A cells or the 468 cells, and you look between 15, 20 minutes or 24 hours, you can see within 2 to 4 hours, these levels of AKT phosphorylation are just really dropping, just as you'd predict. However, in the SUM149s and in the SUM229s, nothing. No effect of the BKM120 on the same target in these two cell lines. Again, they're all P10 null. Very, very surprising. So we said, okay, well, let's just cut to the chase. Let's attack AKT itself. As I said, we've got a drug that turns this off. It's called MK2206. And just as you'd expect in the MCF10A cells, you give the drug, AKT phosphorylation goes away. The 468 cells, you give the drug, AKT phosphorylation goes away. By now, you get the point of the story. Nothing really happens in the 149 to 229 cells are amphiregulin expressing basal breast cancer cells. It's like, this was really a head scratcher. Because now we've attacked directly the target of all of this with, again, a really good drug, at least as we thought, and nothing much happened. All right. So it turns out there was a sort of a key turning point in this story, and it actually came from perusing the Select Chem catalog. And when I did that, and I guess before I say this, let me say one thing ahead of time. AKT actually comes in 3 different flavors: AKT1, AKT2, and AKT3. 3 different genes that make very, very similar proteins that function in very similar ways. AKT3 has not been considered to be a player in breast cancer until recently. I was looking at the Select Chem catalog, and I saw this, and I saw that the IC50, that is the concentration of drug required to reduce the activity of the target by half, for AKT1 was 8 nanomolar. That's a really good drug. That's really potent. But I also saw that for AKT3, it's 65 nanomolar. And when we do this experiment here at 1 micromolar, that's sort of, we choose that dose because that's what you expect to work in this sort of experimental setting for a really potent drug that has this kind of IC50. And it made us think, well, maybe the reason this drug isn't working is because AKT1 is not the target, it's really AKT3, and if that's the case, we're just not using enough drug. Because it just didn't make any sense that the MK wasn't working at all. So sure enough, we went to our RNA data, and here's the 4 cell lines, and here's AKT1, 2, and 3. This is just expression level by messenger RNA. We had this archive data because we've got gene expression profiling and everything. And what you can see is that in the 149s and the 229s, there's a huge amount of AKT3 as compared to the 468s and the 10As. And sure enough, this bore out at the Western blot level, at the protein level. So here's AKT3 in 149s and 229s compared to 10As and 468s. So these cells, in fact, are making a lot of AKT3, and that would suggest that all we have to do now to turn it off is just give more drug, get up to that sort of IC50 concentration. And so we repeated the experiment now. We gave, instead of 1 micromolar, 3 micromolar, 5 micromolar, and now we can finally get rid of the AKT phosphorylation. So that was a really important mystery to solve. Now, unfortunately, there are, as yet, no AKT3-specific drugs in the clinic. It's very tough to make an isoform-specific drug like this. But we're looking for other ways to accomplish this. But this was a very exciting development because it really helps to explain a lot of what we've seen so far. That AKT3 now is the target of all of this, not AKT1. And just like before, I was telling you, almost everything we know about the EGF receptor comes from studying EGF. Almost everything we know about AKT comes from studying AKT1. There's very little literature on AKT3. Okay. So to try and prove that this is really the player, of course, then we go back and we do some knockdown experiments. So before I tell you about that, let me just tell you about one more drug here, this so-called KU drug. This KU drug operates right here in this part of the pathway. This is the mTOR complex. And mTOR is the actual direct kinase that phosphorylates this site. And so, again, this is a pretty good drug. And if you give mTOR to the 10As or the 468s, it's very effective, not effective at all in the SUM229s. But now, if we take these three isoforms and we use shRNA and we can either knock out AKT1 selectively, knock out AKT2 selectively, or knock out AKT3 selectively. And then we can go back and do the experiment with the KU drug. Here's what we find. Now we can get the KU drug to work. By getting rid of AKT3, the KU drug now works. And so does the BK drug. So does the drug that works here. Right? So here's the control. Give the BK drug or the KU drug, nothing much happens. If we knock out AKT1, no effect of the drugs. We knock out AKT2, a little bit of an effect. But if we get rid of AKT3, now these drugs suddenly work the way they're supposed to. So this really pulls the whole story together and suggests that AKT3 is now in the center of this axis, but we still don't really know how it gets phosphorylated, and that's something we're actively working on right now. But here's why this is really exciting as a potential therapeutic strategy going forward. See, as I mentioned at the beginning of the talk, is that gefitinib is a lousy drug in the clinic. It's a great drug in the lab, but for breast cancer it doesn't work. It works in lung cancer if there are EGFR mutations. Why doesn't it work in breast cancer? I really believe that the reason it's a poor drug in breast cancer is because we've looked at the wrong biological effect of the drug. So if you take these breast cancer cells and you give them this drug, they will truly stop growing. They will absolutely arrest and just sit there. But if you then take the drug away, they start growing again. So the inhibition of growth that occurs with this drug in breast cancer cells is completely reversible. And I would argue that reversible growth inhibition will never have any effect in the clinic. If you want to be effective in the clinic, you need to kill cancer cells. And gefitinib doesn't kill cancer cells. And that's shown in this colony-forming assay here. So in these experiments, we expose the cells to the drug for 72 hours. We take the drug away, and we basically ask, what is your ability to form new colonies when we test that? And what you can see right here is gefitinib, even though it can completely arrest the growth of the cells, when you take the drug away, there's no difference in the colony-forming ability of these cells after gefitinib is withdrawn. Again, the growth inhibition is completely reversible. And if we give this MK drug, now the direct AKT attack drug at 1 micromolar, doesn't have any effect. But now, if we combine, and even MK at the 5 micromolar, which is enough to eliminate the AKT3 phosphorylation, even that is not effective at decreasing colony-forming ability. But now, when we combine gefitinib with the higher concentration of MK, now we can really begin to knock out the clonogenic potential of these cells. Now we're getting some real cell killing, sort of a tenfold decrease in clonogenic potential. And that's interesting because we can do the same experiment with these cells where we re-express P10. And I showed you those cells before. This is some 149 cells. Now, if they re-express P10, that by itself, again, does nothing to their clonogenicity. And gefitinib by itself does nothing to their clonogenicity. But gefitinib plus P10 gives you the same effect as gefitinib plus 5 micromolar drug. So this really tells us that P10, the loss of P10, is somehow really important in the expression and activation of AKT3. And putting P10 back gives us a big effect, but we can get the same effect if we can just figure out how to inhibit the phosphorylation of AKT3. And so that's going to be going forward, our goal is to figure out a therapeutic strategy to get this with a better, more effective drug. And again, what we believe is that if we can do that, suddenly gefitinib will be a good drug in the clinic. And gefitinib has been around for a while. It's FDA approved. It'd be very easy to develop clinical trials with gefitinib plus something because we know so much about this drug already. So we're pretty excited about that as a possibility going forward. So this is, the canonical pathway that everybody talks about for AKT3. Unfortunately, that's wrong. And this connection isn't made. We've inhibited this complex, has no effect on AKT3. So right now, we really don't know what the kinase is that phosphorylates it, nor do we know how P10 influences it, but we know P10 does. And so that's the main focus of the work we're doing as we speak. Okay, so in the last couple minutes, I'm just going to talk about some other potential novel therapeutic strategies for this, again, particular type of breast cancer. So I've already talked about possibilities of attacking amphiregulin directly, ways to inhibit Wnt signaling, ways to inhibit AKT activation, which can all be effective. These are all novel strategies, none of which have ever been really tested in the clinic. So are there any others? I think there are. So one of the things that we work on in the lab, and I'm not going to say anything more about how this is done, but we have now with all of our cell lines done what's called a genome scale shRNA screen. And basically this is an experiment that allows us to query the entire genome of our cell lines and ask the question, which genes are essential for your growth and survival? It's a very powerful set of experiments. It uses a library of 81,000 shRNAs that attack 16,500 human genes. It's just really amazing. But when you get the data back from these experiments, you can ask the question, all right, what are the essential genes and where are they within important biological pathways? And so what I'm just showing you here is the result of the screen from the SUM149 cells sort of overlaid on one very well-known biological pathway, the cell cycle pathway. So all of these genes here that are colored in the boxes, all of these genes were hits in the screen. So that tells us that all of these genes are important and essential for the growth of these cells. For many of these, this is not a surprise. This is all the kinds of genes we would expect to find in an assay of this type. But the one that's interesting is this one here, PLK1, polo-like kinase 1. This is an interesting gene for a number of reasons. First of all, it's important when cells are stressed because of their altered genome. It's important for those cells to use this protein to actually progress through cell division and mitosis. And we find this to be a hit in the screen in a lot of our cancer cells. The other reason it's important is that there are now drugs being developed against PLK1. And we do think that, in fact, this is going to be another very good therapeutic target in basal breast cancers. And so based on this finding, we actually looked further at this gene, PLK1, and another gene called BRCA5 that behaved in the same way in this screen. And what we found was really, really interesting. In MCF10A cells, they express PLK1. If you give them this drug, gefitinib, the PLK1 levels drop dramatically, and these cells then die. If you give them gefitinib, these cells will die. These cells won't, but these will. Well, we think we now know why that is, because in the SUM149 cells, if you give them gefitinib, PLK1 levels actually go up. They don't go down, they go up. And these cells, as I showed you, don't die. And the same exact thing is true for this gene called BRCA5, which is otherwise known as survivin, a very well-known cancer gene. You give them drug, levels go down, the cells die. SUM149 say, no, we'll actually make a little more. We're fine. You can keep the drug around all you like. And so we really think that this uncoupling here of the expression of these survival genes, again, is a big reason why gefitinib doesn't work as a drug very well. And so the question was asked then, well, are these good drug targets in these cells? And you can see that because these cells express so much of this, that suggests they really need it to survive. And so if you inhibit it with a drug, you get some really effective cell killing. So it turns out that these SUM149 cells are really sensitive to drugs that target both PLK1 and BRCA5. But then the other cool thing that sort of brings this whole story together is if we now look at PLK1 expression in this experiment with the MK drug and gefitinib, now if we give, again, both drugs together, we can make this look more like that. Now PLK1 levels go down, but only in the presence of both drugs, not in the presence of one of the drugs. And again, we think that's a big reason why they lost their clonogenicity when both of these drugs are present. So the whole thing is really coming together very nicely. We're really making a lot of progress in understanding this altered signaling axis that drives the growth and the survival and the invasiveness of these cells. And at every step of the way, as we learn more about this, we identify other potential therapeutic strategies and drug targets. And again, all of that is will be the emphasis of future work. So the final summary is that oncogenic signaling in AREG-driven P10 negative cells results in activation of AKT3, which is a major driver of drug resistance. Inhibition of AKT3 changes the effect of EGFR-targeted drugs on these breast cancer cells, and that some important genes become uncoupled from EGFR as a result of this AKT3 activation, and some of these genes and proteins are potential drug targets. So it's been really, this has really been an interesting journey so far, and I think there are a lot of potentially exciting things to do next, and we hope to be able to do them. So I'd like to end by just acknowledging the outstanding people that I get to work with every day in my lab. This is my favorite golf hole at Cougar Point at Kiowa. This is the Augusta group that works in my lab, taken at a department Christmas party a couple years ago. So here's Steve Guest. Steve Guest did all of the shRNA screening work that I alluded to. Brittany Ivy is, where's Brittany? She's right there. She works on the 8P11 Amplicon project, which I didn't get a chance to talk about. Christy Kapler is here, and Christy, along with Erica, who's there, do all the experiments on the P10, the amphiregulin, the AKT3. This is Alex, who works, again, on the 8P11 Amplicon project. Zach works with Steve. And Ginny keeps me on time and focused, and keeps everything, keeps the trains running on time, I guess. So thank you all for your attention. I'd be happy to answer any questions. So there are a number of potential mechanisms in these particular cells. There's actually a deletion in an intron in the P10 gene that causes the message not to be truncated and then degraded, and so there's no protein made at all.